Can you hear me? All right. So today I'll talk about mitochondria and the demise of neurons. And the question that we set out to answer a number of years ago was, what are the molecular mechanisms that are required for the survival of neurons in adult fly eyes? And when you look at the literature, you realize that there is a lot of different components that have been implicated in the demise of neurons: oxidative stress, dysfunctional autophagy, endolysosomal function, the mitochondria, misfolded proteins, etc. So there's a lot of different mechanisms to kill a neuron, which is not unsurprising, I would say. But very often we don't know what the primary causes are that lead to the demise of neurons. And what we set out to do was to look really early on. And that's what the screen was. First was to create mutations in essential genes that, when muted in the photoreceptors, lead to a gradual demise of the ERGs, the electroretinograms. And that then led to an EM screen. And that EM screen was done in very young animals prior to the onset of symptoms. And that's where we really got a lot of information. And I'll try to make that clear later. So then we focused on the molecular and cellular mechanisms. And what we found in many cases is that there's probably very often synergism. You're all familiar in cancer biology that there's the two-hit hypothesis. And we think that this is about the same for neurodegeneration, is that very often you have an insult and then that doesn't do a hell of a lot, and then you have another insult, and it's when those two come together and synergize that you get a severe defect. So the screen that we set out to do was an ERG-based screen where you poke an electrode in the eye, an electrode in the thorax, shine light, and measure the electrophysiological extracellular recording. And this is a typical electroretinogram, and there's several types of neurodegeneration. There's those where the ERG essentially becomes a flat line in the old flies. And there are those where you lose these on and offs. And these on and offs are indicative of neurotransmission between the photoreceptor and the postsynaptic cell. So what we did is we used induced mutations with EMS, ethylmethane sulfonate, on one chromosome, and on the other chromosome we put a cell lethal mutation, so that when you use the eyeless flipase in the eye and you make progeny of this cell after DNA replication, you're homozygous for the mutation in one cell and you're homozygous for the cell lethal mutation in the other cell. Therefore, you essentially eliminate this wild type cell, and you give a better chance for the cell that carries the mutation to expand and make bigger clones. And then we first record in old flies and see if we see in old flies. As I said, these genes are essential, so we first took created 35,000 stocks with mutations with very low concentrations of EMS, and just screened for those that were lethal, which was 6,000 of them. And then we performed these ERG screens. And once you've identified mutations like this, or even if they're much less severe but show that profile, we perform electron microscopy. And as I said, this has been the most valuable information for us, because it's really led many of the projects, as you will see, in the lab. So you cut and make a section through the retina of the eye, or through the lamina, which is where the synapses are of these photoreceptors. And when you do that in the retina, you see the rhabdomeres of the photoreceptors. But you obviously have all this ultrastructure where you can look at the ER, and you can look at the mitochondria, and you see the lysosomes, and you see the Golgi, and you see all these ultrastructural features that are really important, because when we do this in an early age and we see particular defects in some of these organelles, we immediately get a hint as to where the problem starts, because this is essentially at the onset or before the onset of the phenotype. We also look at the synapses in the lamina, and here you see the 6 photoreceptors in a cartridge. So there's 6 photoreceptor terminals here. these are the postsynaptic cells, and the black area here is the glial cells. And so the glial cells insulate each of these cartridges. And again, you can look at a lot of ultrastructure. Typically, there is a single mitochondria in a single section. There's captate projections. There's active zones that synapse with these postsynaptic cells. And again, you have a lot of ultrastructural morphology that is really important. So now if you look at the genes that we've isolated, we isolated 700 mutations that cause an age-induced retinal degeneration. We did this screen on the X chromosome, and as I said, we did a very extensive screen. And so typically we isolated about 4 alleles per gene. So this is not 700 genes. This is 700 mutations corresponding, as you'll see in the next slide, to about 160 genes. If you look in the young flies by EM, and this is a mutation in MARF, that is the fly homologue of mitofusin. It causes Charcot-Marie tooth disease in patients, in human patients. You can see that there is an expansion of the ER, which is shown here. There are cells that really look already sick and have accumulated all kinds of vesicles. And there are lipid droplet accumulations in the glial cells here that you can see. So there's various pathologies, even though these photoreceptors are still quite functional. They're not normal, but they're quite functional. But if you record at this stage, you get almost nothing anymore. And that's pretty obvious. The photoreceptors are completely disrupted. But note that they're not gone. Most of the mutations lead to a demise of neurons. It's very gradual. And apoptosis only kicks in very late in most of the mutants. Not in all, but in most. Here's a mutation in Sicily, and the human homologue of this gene causes Lee syndrome. And again, at the young age, in the young flies, you see that there are defects, and there's accumulation of lipid droplets in this mutant, and the photoreceptors are kind of squashed together because there's an expansion of the glia. This causes a very severe disease in infants, and patients with mutations in this gene are all dead by the age of 1. Here is another gene that causes Lee syndrome, where they live a little bit longer, LRP-PRC, and here we see a loss of photoreceptors. And again, if you look late, there's very little information to be gained. If you look at a patient that dies, and you look at the morphology of the neurons, I argue that there is very little information to be gained there, that you really need to do this at a much earlier step, which is obviously problematic in humans. Now, when you look at the genes that we've identified, so all of these genes were mapped by duplication mapping, and then the whole genome was sequenced, and then each of the genes that is listed here was rescued with Pacman clones. So if you look at the list, you see that many of the genes that are highlighted in yellow here were already shown to be disease-causing genes in humans. So about 30% of the genes are genes that have mutations in human genes. Many of these are neurological syndromes, but not all of them. More than 93% of these 165 genes have human homologs, and that's a lot more than the typical Drosophila. About 60% of the genes in Drosophila are conserved, but here from this screen, we ended up with about 93%. So there's a number of issues that I want to point out here. If you look at this list here, for example, this is para, which is the sodium channel. In vertebrates, there's a lot of sodium channels. And you can see that of the 11 sodium channels that have been identified, 9 are known to cause human disease. And in fact, this is a recurrent theme. Every time that there are more human homologs than a single one for these genes, like Notch or Schlank or little one myospheroid, you always have human disease. But when you have single genes, very often, there are no human disease. Sometimes there are, but that's not the majority of the cases. And so this is an interesting observation, which is kind of summarized in this little table here, where we have 165 genes, and there's 12 genes that don't have a human homologue. There's 96 genes that have a single human homologue, and there's 57 genes that have more than one human homologue. And now, when you look at this, you see that for these 96 fly genes, there's only 21 human disease. That's less than 20%. But for these 57 genes that have more than one human homologue, there's 100 diseases in the OMIM database. So this argues that an essential gene in the fly is probably essential in the human, and is not a very good candidate for a human disease, so that only very small and subtle mutations in those genes are going to manifest themselves as phenotypes. Most of these are going to be abortions. But when you look at these where there are multiple homologs, then there is redundancy built into the system, and they become better candidates to become human disease. So this is important because even if you correct for the number of human homologs, you still have a threefold enrichment. So this is kind of an interesting observation because we use this now essentially to drive a gene knockout project in the lab, where we focus mostly on genes that have more than one human homolog because they are low-hanging fruit and they will help us in the identification of other human disease. Now, I mentioned when I showed you the table that there's the yellow genes, and these are obviously interesting because many of these did not have mutations, and so they allow us to characterize these mutations better and try to understand the pathology of the disease. But there's all these white genes, and so can we use these white genes to discover new human disease? And for that, we teamed up with Jim Lupsky at Baylor College of Medicine and Richard Gibbs. And in the Mendelian Genomics Center, what they do is they sequence patients. So a lot of patients go to a clinic, are undiagnosed, and when they're undiagnosed and the physicians or the patients get frustrated, they can send a sample of their blood, and they'll sequence at Baylor their genome, and this is a collaboration between Baylor and Hopkins. Currently, there are 7,000 individuals that have been sequenced, but when we initiated the project, there were about 2,000. And we looked at those 2,000 genes and looked at the genes we had identified in Drosophila, and then went back to that database and tried to identify mutations in genes that could be causative for human disease. And obviously, you can separate these in recessive and dominant models. And we found 8 candidate genes here with Mendelian expectation, and we found 15 genes for the dominant model. And we solved 2 cases here for CRX and DNM2, but I'll just give you this example and show you how we did this. Now, one of the major issues is when you have a single individual, you have so many rare polymorphisms that it's very difficult to really define what the causative mutation is. And so here in this case, we have a patient, a single individual that was sequenced, and these are all the possible candidate recessive mutations in a typical patient. This work was done in Jim's lab, and they looked at a lot of cases, and this is a summary of a bunch of cases. So there's typically about 50 rare polymorphisms that could be causative for a disease in a recessive model, and typically 150 rare variants if you go for a dominant model. Now, if you sequence a second individual, a father or a mother, you reduce that, obviously, and if you have a trio, you're down typically to this range. And this range can be narrowed down further because typically you can use bioinformatic programs and reduce the number to typically 2, 3 variants, sometimes 1. If you have 4 individuals, you're in better shape. If you have 6 individuals from the same family, you can almost invariably identify the mutation unambiguously. But we don't operate most of the time in this because it's very difficult to get 6 members of the same family since these are really rare disease. So we typically operate in this range here or in this range here. Now, one of the genes we identified in the screen was this gene here, which is called ANKL2. And when we made clones for this gene in various tissue, we saw that there was a loss of neurons. So this was not a degeneration mutation. We actually screened for 20 different phenotypes. We invested so much time and effort in creating all these mutations that we thought we should get a very broad database and collect not only neurodegenerative mutations, but also neurodevelopmental mutants. And so this was a gene that we had identified. Almost no data published on this particular gene. It has 3 domains. And we found a patient, this kid here, that had a truncation. So this is a recessive model that had a truncation here and a point mutation here for an L to V. And when I saw this, I thought, wow, that's really a weak mutation, right? An L to V, that's not much of a change. Now, this boy here had a sister, and his sister died after 3 weeks. The boy is still alive now. He's 6 years old, but he's 16 standard deviations below the mean in growth. He weighs about 10 kilograms and is 6 years old. And when you look at the brain, you see that he has very severe microcephaly. In fact, there's so much skin that there are folds of skin on top of his head because of the microcephaly. So here you have a very severe case of microcephaly, and the question then is, are these mutations really the cause of this microcephaly? You only have a single individual. So we turned to the fly and started looking in the fly and compared the brain in the third instar larvae of ANCO2 mutations with control. And we then ubiquitously overexpressed the human gene, ANCO2, and fully rescued the brain phenotype that we observed, the small brain phenotype. And in addition to rescuing the brain phenotype, also rescued the lethality associated with the loss of this mutation, and that's shown here. We then looked a bit more carefully to try to understand what's happening in these mutations, and essentially these animals develop perfectly fine to the early third instar larval stage. And when they reach the early third instar larval stage, brain development starts to arrest, and you make less stem cells, neuroblasts, when compared to wild type. The neuroblasts often divide symmetrically, and that symmetric division leads to an aberrant differentiation of the neurons. When the neuroblasts divide and are formed, they form many fewer neurons than the wild type neuroblast. This is the progeny of the single neuroblast, and here the progeny is very little. And then there is massive apoptosis of those neurons. So you have a combination of defects of lack of specification of neuroblasts. You have a problem in the division of neuroblasts and asymmetric localization of key determinants, and then you have massive apoptosis. So this is just one case, and then further in the talk I'll talk about some other cases. But this work was done by Manish and Shinya, and in essence we've been working on a number of genes, actually 23 genes, that allows to study these genes in Drosophila that cause human disease. And the reason why we focus on this gene is RNAi against these genes barely has a phenotype. So this is another feature that came out of the screen that I'm not going to show you, but other screens have been done, neurodevelopmental screens for loss of neurons in the fly using RNAi, and there's essentially no overlap between the RNAis that have been identified and the genes that we've identified in our screen. There's less than 10% overlap. So this gives us a very nice set of genes to work on. The fact that we can discover new human disease, and I'll show you a couple more cases later in the talk, and this observation that I mentioned, that there's only 3,500 genes in flies that have multiple human homologs. And that is really low-hanging fruit because many of those, as I've argued, will allow us to study human disease. The X-Screens team is a large team of people and graduate students and postdoc in my lab that all really collaborated together and did a wonderful job, and they were led by Shinya and Manish in my lab. Now, one of the key features that came out of the screen, which kind of was surprising to me, but may not be surprising to MDs, is the fact that a lot of the genes that cause neurodegeneration, there were about half of the genes we identified cause neurodegeneration and code mitochondrial proteins. So 40% of the genes that cause neurodegeneration in the screen correspond to mitochondrial genes. And then there's about another 20% that correspond to endosome, lysosomal, autophagy genes, and the other pool is pretty heterogeneous. Now, when you look at mitochondrial genes, people typically think that the mitochondrial genome is the key factor here, but really the mitochondrial genome only encodes 1% of the proteins that are present in a mitochondria. 99% of the mitochondrial proteins are encoded in the nucleus and therefore would be subject to the mutagenesis. Studying these genes is very tricky because the genetics here is very hard to do. Now, mitochondria you typically think as ATP production machines. At least that was my impression when I started working on it. But if you dig into it, you'll find out that they do a lot of different things. Everybody knows that they're involved in apoptosis, but they control cell proliferation, and Utpal Banerjee here has shown that steroid synthesis, iron sulfate cluster synthesis, pyrimidine synthesis, calcium buffering, energy and heat production. So they're really involved in the different processes, and it's not surprising, therefore, that mutating different genes in the pathway may lead to very different phenotypes. And so we see phenotypes where the synapses degenerate first and the cell body is intact, and we see mutants where the cell body is degenerated and the synapses are intact. So you get all kinds of different combinations depending on what gene you mutate. And I'm going to tell you a short story from one of the graduate students in the lab. And this story started, again, by doing transmission electron microscopy. And this story is quite exciting because what Lucy has shown is that lipid droplets accumulate and presage neurodegeneration in a subset of the mutants. So what we first tried to do was to look at a lot of the mutants and try to kind of look at the mutants and see if we could see common underlying phenotypes. And here, Cicely, Marv, and Atzmet, 3 genes have a very similar phenotype early on. So if you look at the control animals and you look at the photoreceptors, you see that the glia, this blue cell here, which is a pigment cell, plays an insulatory role for the photoreceptors. And if you lose this glia, obviously the photoreceptors are going to be severely affected. Now in Cicely, Atzmet, and Marv, and I'll come back what these mutants are, the glial cells are vastly expanded in size, leading to a kind of a squishing phenotype, because the glial cells expand and the photoreceptors kind of squish together. And that's what you see here, also a little bit here. And they're loaded with lipid droplets. And this is true for these 3 mutants, and this is an all or none phenomenon. They have lipid droplets or they don't. In fact, wild-type animals, we've never seen lipid droplets. Now, we had to show that they were lipid droplets, and so Lucy developed an IL-red staining protocol, and in that protocol, you can clearly see that the photoreceptor rhabdomeres, which is a stack of 5,000 membranes piled on top of each other, are positively stained, but the glial cells here, which surround this, are not stained. But in Cicely, Atzmet, and Marv, you see the rhabdomeres, and then there's all this ectopic Nile-red staining, which are lipid droplets that are present in glia. And this is also true in Atzmet, and this is also true in Marv. In fact, she knocked out other genes that are mitochondrial genes that show a similar phenotype. ND42 is a gene that is a critical component of complex 1 in the electron transport chain, and that also leads to lipid droplet accumulation. In fact, an RNA knockdown of Parkin also does that, but not PINK or DRP. In fact, the number of mitochondrial genes that cause this phenotype can be easily discovered by just doing a simple assay, reactive oxygen species. If the animals have high levels of reactive oxygen species, we have a lipid droplet accumulation. Anywhere below a certain threshold, there is no lipid droplet accumulation. What's interesting is that this is an early phenotype. So you see these lipid droplet accumulations here in a young animal. 3 days later, there are many fewer, and if you go later, when the photoreceptors start to degenerate, you barely see any lipid droplets. So this is really an early hallmark of the initiation of the process of neurodegeneration. So what she came up with, Lucy, after a couple of years of work, was to show that when the mitochondria have a very high level of reactive oxygen species, they activate a kinase called JNK, and JNK phosphorylates SRABP, and SRABP is a key factor in lipogenesis. And what happens here is that SRABP will lead to the production of a lot of lipids, but they never stored in neurons. They're immediately transferred to glia, and there they pile up as lipid droplets. So they pile up in these lipid droplets, and these cells expand. And because there is a very high levels of ROS, there is massive amounts of peroxidation. And I'll show you that. And that massive amount of peroxidation in glia, which we do not observe in neurons, makes these glial cells dysfunctional. And that obviously leads to a negative feedback. And I'll show you that this is not only true in flies, but that is also true in mammals. And so the genes that are involved in the process here have 3 very different genes. Usually as geneticists, when you identify a gene and a phenotype, you think that they function in the same pathway. They have the same phenotype, and typically, if you look at the wingless pathway or notch pathways, the genes in those pathways have similar phenotypes. Here, that was not the case at all. So here you have Cicely, which is a chaperone for complex 1. Marv, which is involved in mitochondrial fusion, and Atzmet, which is involved in the translation of the 13 mitochondrial transcript. This gene causes Lee syndrome, it's recessive. This gene causes Charcot-Marie-Tooth disease and is dominant, and this causes a recessive ataxia. So very different pathological, essentially, phenotypes associated with the loss of those proteins. As I said, in Lee syndrome, there's lactic acidosis and rapid neurological decline, whereas here the onset depends on the nature of the point mutation. I should say that there is a autosomal recessive form of CMT in MARF, but the kids die at birth and can't be saved. Whereas in this, mutations in this gene are very peculiar because they're very partial loss of function mutations or aberrant duplications of the gene. So these proteins, as I said, play very different roles. Sicily is the chaperone for complex 1, whereas morphin mitofusin plays a role in mitochondrial fission and mitochondrial axonal transport, and ER mitochondrial anchoring. And this is in methionine tRNA synthetase that loads a methionine on the proper codon during translation for the 13 mitochondrial protein. What these all share is a very severe level of ROS. This one is slightly less and also has less lipid droplet accumulation. And the work on the function of those proteins was published earlier. As I said, lipid peroxidation is really important. And here you measure peroxidated lipids with a bodipid dye, and you're looking in the control. There are very few lipids. There are some lipids in the glia, but very few. You look in Sicily, MARF, or ATSMET, where the photoreceptors are, there's no lipids and there's no lipid peroxidation, but where the glia is, there is massive amounts of lipid peroxidation. And this is true for the 3 mutants. If you overexpress SRABP, you can induce massive amount of lipid droplets. They're not pathogenic, they don't do anything, but there's also no ROS, and so they don't show up here. And so there are no peroxidated lipids. Accumulation of lipid droplets is not pathogenic. When I presented this data at the NRI in the institute where I work, Huda looked at me and said, Hugo, this is not being documented in vertebrates and is not involved in the pathogenesis in neurological diseases in vertebrates. So I took up the challenge and called Richard Palmiter, who'd made a knockout for a gene that is a subunit of complex 1 and was shown to cause Lee syndrome. And they made a knockout for NDUFS4, and the first thing Lucy did was to knock down this gene in Drosophila and showed that there was lipid droplet accumulation. And again, the lipid droplet accumulation only occurs if you knock out these genes in neurons, not if you knock them out in glia. In all cases, you need to knock down the genes in neurons to get the phenotype of lipid droplet accumulations in glia. So these mice are interesting. They're pre-symptomatic at day 23. They show the first sign of ataxia at day 34. They're very ataxic at day 50 and need to be sacrificed around day 55. When I called Richard, his postdoc, Alberto Quintana, who did this work, I asked him to send us brain, pre-symptomatic brain, so when the mice were still normal and had no obvious phenotypes. And Lucy came back after 1 day doing dissection and showed that there were massive lipid droplet accumulation in 3 areas of the brain. And that was interesting because when I called Richard and I said, there's 3 areas of the brain where we see lipid droplets, he said, Hugo, that's the first 3 regions in the brain where we see neurodegeneration. So there's a perfect correlation between where the lipid droplets accumulate and where the onset of the symptoms is. And like in the fly, the junk kinase phosphorylation is increased. And so to show you where these lipid droplets accumulate, Lucy did a nice staining, and you can see the lipid droplets are shown in yellow here, and the blue cells are microglia, and the red cells are astrocytes. And this, in these 3 areas of this brain, this is the hallmark of the pathology here. You have an accumulation of lipid droplets in the microglia and in the astrocytes. And again, this is pre-symptomatic. This is actually in mice that are just born, before the onset of symptoms. So the question then is this really, this phenomena of high ROS, is this really what's causing this accumulation of lipids and the phenotype? And we showed this by feeding an antioxidant. We'd shown in the fly, Lucy had shown in the fly, that feeding an antioxidant was a very nice potent suppressor of the degenerative phenotype and delayed the onset of degeneration by 5 to 10 days. This drug is very expensive and costs $14,000 for a gram, so we didn't do a long time experiment. we did kind of 10 mice and injected them for 1 week at the onset, prior to the onset of symptoms, and then did a rotor-rod assay and compared controls with the knockouts, which, as I said, lose their ability to stay on the rotor-rod after at day 34 already. In this, we tested at day 30, but a single injection of daily of AD4, which is a potent antioxidant, nicely suppressed the phenotype. We've now done it for 2 weeks, and actually for 2 weeks it works as well. And it's interesting because it prolongs and delays the symptoms exactly by the amount of time we inject the drug. So there is a delay here in survival over the entire length of the life of the animal, and they live a week longer and have an onset of severe symptoms a week later. And if you feed them for 2 weeks, the same thing happens. They live 2 weeks longer, and they have a better performance on the rotor rod. So I think you're now hopefully convinced that this process is involved in neurodegeneration. The question now is, how do you transfer lipids? How do you accumulate these lipids? And what are the molecular mechanisms here? And Lucy started looking at the neuron glial lactate shuttle hypothesis. The idea is that the blood is here in the vertebrate, and the glial cells, obviously, surround the blood vessels and take up the glucose and turn it into pyruvate and produce lactate that they then secrete in the extracellular space, and the lactate is taken up in the neurons to form pyruvate and drives the energy cycle in the cells. And she's done some very nice work, and I'm going to just summarize this because I want to switch to another story. But essentially what she's found is that lactate that is produced in the glia is transported through a monocarboxylate transporter in the extracellular space, and that lactate is taken up by another monocarboxylate transported into the neuron. And this is really the energy source of the neuron. This is where the neuron derives almost all of its energy from. Lactate dehydrogenase then produces pyruvate, which through pyruvate dehydrogenase produces acetyl-CoA, which drives the mitochondrial energy cycle. In the mutants that we're looking at, this acetyl-CoA cannot be consumed in the TCA cycle because there is a problem in the function of the mitochondria. And therefore, citrate is formed, which is then used in the lipid synthesis pathway outside the mitochondria. And then enzymes use this acetyl-CoA that they have to make fatty acids and lipids that are transferred to this fatty acid transport protein, and then they're captured by apolipoproteins, a neuronal apoprotein. This apolipoprotein is secreted by neuron, and this apolipoprotein is secreted by glia. To take up into the glial cell to the same fatty acid transport protein, the lipids, to form the lipid droplets. And disrupting these pathways allows you to suppress the lipid droplet accumulation. If you kill this, or this, or this, or this, or this, you always suppress lipid droplet accumulation, and in all cases, you delay the neurodegenerative phenotype. So I think I'm not going to continue to elaborate on this, but this is a really interesting protein because in vertebrates it's ApoE. And ApoE is the protein that causes Alzheimer's disease. And we've tested and shown that we can completely replace this protein with ApoE. But ApoE4, the mutation that's implicated in Alzheimer's disease, is deficient in this process. And so this brings us to starting to be able to study the molecular mechanisms associated with ApoE dysfunction in Alzheimer's disease. I'm going to end with a third story from one he in the lab on a protein that you probably never have heard about, nardelysine, which we identified in the screen, and found that this protein is actually a protein that was not known to be in mitochondria. And when he showed that this protein is in mitochondria, and it leads to a very specific defect, namely a dysfunction of a single enzyme in the TCA cycle. And the dysfunction of that enzyme in the TCA cycle dramatically affects autophagy. And that is an unsuspected observation. And this is true in flies, mice, and humans. So the screen, as usually, starts by doing ERGs. And I just wanted to show you the phenotype so you have an idea as to what the phenotypes are. These are young nardelysine mutations. We always do a genomic rescue to show that the mutation is really due to the gene that we've identified. And you can see here that in these mutants, there is a reduced depolarizations of the ERGs, but the ons and offs are fine. when you look at day 35, you see that there is a loss of the on-offs. Now, this is a very slow neurodegenerative phenotype compared to the ones I've shown you before. The genes I've shown you before usually are very dysfunctional after 10 or 15 days. But this, you see no phenotype till day 30, and only after day 30 do you start seeing a degeneration of the neuron. And when he quantified these defects, and that's what these ERG defects are. Upon identifying the gene, we found 4 mutations in nardilysin. They're all stop codons. And this protein, this gene encodes a protein that has an insulase domain and a substrate binding domain. And just to show what the phenotype is that's associated with these various mutations, using light microscopy at day 1, the nardilysin mutants are just like wild type, but if you look in 35-day-old animals, you clearly see the vacuolization, the loss of photoreceptors. And there's also a defect at the synapses. The synapses are vastly expanded. So this is a single synapse in the lamina here, in one of the cartridges. And you see how large these synapses are. And they're jam-packed with vesicles and autophagic phenotypes. So there's, again, a very slow progressive neurodegeneration that's actually more prominent in synapses than in the photoreceptors. When we dig in the literature, we found that this protein is a zinc melanoendopeptidase, that it can cleave several neuropeptides, and this was done by biochemists, and then some showed that it localized to the plasma membrane and functions as an activator of ADAM proteases. Others argued that it's localized in the nucleus and plays a role in transcriptional regulation, and finally nardilysin was shown to be present in the cytoplasm and played a role in antigen processing. So very different functions associated with this protein based on biochemical data and some cell biology data. Now, a team in Japan, Ono et al., made a knockout, and this led to an interesting observation, namely that 80% of the pups that are homozygous for a complete knockout of nardilysin die around birth or live for a few days. But 20% survive over the entire lifespan of the mouse and live for 2 years. But they develop a microcephaly. So over time, their brain becomes smaller and smaller, and there's progressive loss of neurons, and they undergo a neurodegenerative phenotype, and they lose the corpus callosum. Now, when we were looking at this protein, the first thing one he did is it looked carefully at the homologs and immediately noticed that something weird was happening. A, the gene is conserved in evolution from flies to man, but they all have a mitochondrial targeting sequence that was completely ignored in the previous literature. And so this suggested that this protein really should not be where it's supposed to be based on the previous literature, but that it should be in the mitochondria. So we had obviously to test that this mitochondria was targeted to mitochondria. And that's what's shown here. He expressed in S2 cells a wild-type protein, and it perfectly colocalized with mito-GFP. If you remove the mitochondrial targeting sequence, you get nuclear localization, you get cytoplasmic localization, and you get membrane localization. And this is true in vivo, and if you remove the enzymatic activity and make a single amino acid chain, the protein is also mislocalized. So we were excited. We turned to the mitochondria and started putting all our battery of mitochondrial assays that we have available to study the function of this protein. Protein distribution that we can look at is perfectly normal, and ADH levels look normal. Mitochondrial DNA copy looks more or less normal. Mitochondrial potential looks normal. ATP production looks normal. The only thing that we noticed was that there was an elevation of the electron transport chain activity for complex 1, 3, and 4. So this is really a relatively mild defect and really not easily explain what's going on, and certainly doesn't explain the pathology. So with this in mind, we started looking for patients that have mutations in this gene. And we were actually lucky and connected with UCLA through Valerie Arboleda and Dr. Nelson, and identified a patient that is homozygous for a stop codon in this gene. And this is a relatively large family of a father and a mother that each are a carrier of this mutation since the kid is homozygous. We don't know what happened to this kid, but apparently it died near to birth, and then all of these others are carriers or homozygous wild type. And the phenotype is pretty severe. It's a 14-year-old male and developed an acquired microcephaly with severe intellectual disability, doesn't speak, has severe hypotonia, is unable to walk, is ataxic, is wheelchair-bound, and is on a liquid diet. And here is what you see for the patient brain. You see the microcephaly. So the first thing we did was to rescue the fly gene mutant with the human cDNA, since we only have a single family, and again, we have a single affected individual. And as you can see here, there's a full rescue of the neurodegenerative phenotype and a full rescue of the physiological function. The on and offs, as measured here, when rescued with the human cDNA, go from this to this at day 35, and from this to this when we overexpress the human gene. And the point mutation in the patient is actually between two stop codons that we identified, and so obviously this doesn't rescue the phenotype. So now we're kind of stuck with the fact that we have a potentially interesting gene. It causes a microcephaly in mice, it causes a microcephaly in human. It's probably a mitochondrial protein. What's going wrong in the mitochondria that caused this defect? And so we turned to metabolomics to answer this question because we couldn't find any other defect. And we're struck by the fact that two compounds, alpha-ketoglutarate and glutamine, were dramatically upregulated in the mutants. So we called the Japanese team, Ono and his colleagues, and asked us to send mouse embryonic fibroblasts and perform the same metabolomic analysis. And guess what? Those two compounds, alpha-ketoglutarate and glutamine, are upregulated in the mouse embryonic fibroblast. So now we have to rethink and focus on something else, and that is the TCA cycle. So now we're in the TCA cycle, and in the TCA cycle, alpha-ketoglutarate is upregulated and glutamine is upregulated. Alpha-ketoglutarate is the substrate for alpha-ketoglutarate dehydrogenase, and this would predict, obviously, that this enzyme may be deficient in its function. And that's indeed what I'm going to show you. OGDH is defective, and therefore there is a pileup of alpha-ketoglutarate, and that has severe consequences. So the first thing that he needed to do was to do an assay and show that fly OGDH activity is decreased. And indeed, you can see that fly OGDH enzymatic activity in these mitochondria is very severely decreased, whereas isocitrate dehydrogenase, citrate synthase, and malate dehydrogenase activity, all components of the TCA cycle, are functionally normal. And when he tagged OGDH with GFP, and looked at the stability of the protein in NRD mutants, you can see that, in fact, the protein is lost in cytoplasm and in mitochondria. So there clearly is a component of this protein that's present in the cytoplasm, and there's a component in the mitochondria, and both are lost in this mutant. So this was interesting, and then we said, let's look at the mouse embryonic fibroblast cells and see what we see there, and we were surprised to see something different. In mouse embryonic fibroblasts are null for NRD, and so there is no protein. But look at what happens with oxyglutarate dehydrogenase. There is a vast upregulation of OGDH. So we thought, wow, that's really weird. So we measured the enzymatic activity, but this protein is barely enzymatic active. It's the same as in the fly. So in both cases, you lose enzymatic activity for oxyglutarate dehydrogenase, but in one case, in the fly, you lose the protein, and in the MEF cells, you pile it up. And when I saw this data, I discussed this with Juan, and I immediately thought there is a problem with the folding of the protein. The protein is probably folded improperly, and that's what at the cause of a pileup of dysfunctional protein or the loss of the dysfunctional protein. I should say that mitochondria have a very specific pathway to deal with misfolded proteins. Misfolded proteins in mitochondria are cut by peptidases and cut into 15 to 20 amino acid domains and exported outside the mitochondria. So there's no ubiquitination, there's no autophagy pathway in the mitochondria. They're just exported through the mitochondria into the cytoplasm and eliminated either through autophagy or proteasomal degradation. So what I've shown you so far is that in the TCA cycle, nardilysin is doing something to OGDH, and how can we figure this out? So what he did is he tagged NRD1 with GFP and did this mass spec, this immunoprecipitation with these nano antibodies, these LAMA antibodies that are really efficient at pulling down 95% of the protein, so you can do a nice mass spec analysis. And out of the mass spec came some interesting data, namely that first, yes, it binds, nardilysin binds to OGDH based on this assay, and we've verified that with other assays. But then notice what comes down: mitochondrial chaperone, mitochondrial chaperone, mitochondrial chaperone, mitochondrial chaperone, mitochondrial chaperone, mitochondrial chaperone, etc. So a lot of mitochondrial chaperones. And as many of these chaperones are actually interesting proteins because they not only function as chaperones, but they have peptidase function. So they're the two functions of the protein are kind of integrated in a single protein. It tries to fold. If it's misfolded, it'll cut the protein. And so that's what you see here. There is this protease activities that's present in several of these chaperones. So what this then suggests is that nardilysin binds to OGDH, and what it does is, together with all these other chaperones, ensures that the pre-protein is properly folded and stays properly folded, because it also binds to HSP60, and ensures that this protein is active. And in the absence of nardilysin, there is a problem with OGDH, and then there is an accumulation of alpha-ketoglutarate. But there would be another possibility. Maybe nardilysin would be required for the import. And if it's not imported, you accumulate the protein in the cytoplasm in vertebrate cells or in Drosophila. You get stuck outside the mitochondria. So when he wanted to show that the protein is imported in mitochondria and did a nifty, simple assay, he purified mitochondria and then purified OGDH that was in vitro translated, and then for the indicated time points looked at the transfer from the soluble fraction from the cytosol or the medium into the mitochondria. And these are all purifications of mitochondria, and you can see that indeed in wild type, the wild type being the mutant with a genomic rescue construct, the OGDH is imported into the mitochondria. In nardilysin, it is important, and then it's degraded. It can't hang around. So we then showed with another assay that the protein was indeed misfolded, and the way he did that is he purified mitochondria and looked in cells at the supernatant and the pellet, and then used labeled OGDH, and then raises the temperature at 42 degrees and looks at the supernatant and the pellet, and most of the protein precipitates in the pellet and cannot stay in the supernatant upon a heat shock, whereas other proteins don't show this phenotype. So this suggests that the protein is indeed misfolded. So now we have this cycle, and we think we've provided evidence for a chaperoning activity of nardilysin by recruiting chaperones to make sure that this protein is properly folded. And we went to the database that I mentioned before from Jim Lupsky and Richard Gibbs and looked for patients with mutations in OGDH. And we found this patient. The girl is 15 years old. Here's the tree. It's a recessive again. This lady here had a brother that lived for 30 years and had an acquired microcephaly and just progressively got worse, just like she has been getting worse, and developed also a very severe microcephaly over age, but it really took 30 years before he died. And this girl, as I said, is 15 years old, also has a microcephaly, and has a very similar phenotype to the nardilysin patient that I just showed. So there is hypertonicity, an unremarkable prenatal history, just like the nardilysin patient. There is no ambulation. She's wheelchair-bound. She cannot speak, and she's fed with a liquid diet. So these two patients share very similar phenotypes. Now, the key issue here is, here we don't have a stop codon. We have a single amino acid change, and we need to show that that amino acid change is really responsible for a loss of function of OGDH. I should say this is OGDHL and not OGDH. This is OGDHL is mostly expressed in the brain and the liver, whereas OGDH is expressed in all the other cells in the human. In flies, there is only one homolog. So this is the point mutation. This amino acid is conserved between OGDHL, OGDH, the fly OGDH. It's in a very conserved domain of the protein. And you can see that this is the amino acid that is changed here. And as I said, this protein is highly conserved. So now a few words about Drosophila biology and methods to test point mutations. we developed a kind of a pipeline to test human mutations. And so if you identify a mutation in a gene that is not a stop codon using whole exome sequencing or whole genome sequencing or GWAS, you identify a gene, you then use an orthology prediction program and find the fly gene, and this works for about 60% of all the human genes, in fact, slightly higher. Then we take the fly gene and we kill the fly gene by putting in this cassette, which contains a splice acceptor T2A gal4 poly-A tail. What this does is there's a splice donor here that goes to the splice acceptor that forms this protein and that forms this transcript, and the transcript is aborted at the poly-A tail, and so it truncates. And then this portion here, the T2A, blocks translation at this level, reinitiates translation at the gal4 and produces the gal4 in a proper spatial and temporal expression pattern. And then you can now drive a human UAS cDNA that you put in the fly to rescue the fly gene and essentially humanize the fly for that particular gene. And so using this approach, when he first tried to rescue a null mutation for OGDH in the fly and failed, but when he was able to put in this construct and produce the gal4, he was able to show that the UAS fly cDNA was able to rescue the lethality and the phenotype, that the fly point mutation failed to rescue, that the human OGDH was able to rescue, and that the human point mutation failed to rescue. And this in the longevity assay and in the behavior and in the ERG assay. So this really shows that this mutation is pathogenic. In the last two slides, I want to come back and talk about the mechanism by which this gene could cause this neurodegenerative phenotype. So I've shown you that there's high levels of alpha-ketoglutarate, 3 to 8 or 9-fold the normal levels because of the loss of OGDH or nardelysine. Alpha-ketoglutarate can get out of the mitochondria and has been shown to activate TOR signaling. And obviously, if you activate TOR signaling, you suppress autophagy. And the idea is that this is a pathogenic pathway that is involved in the process. So can we show that TOR signaling is upregulated? And indeed, if you look in nardelysine mutants or in the OGDH knockout, you see that there is an upregulation of S6 kinase, there is an upregulation of 4-ABP, the same is true in OGDH, and then there is, so this argues that the TOR signaling pathway is upregulated, and then there is a problem in an accumulation of P62, and an upregulation of P62 suggests that there is a problem with the degradation of proteins in the autophagic pathway. And so then we thought we should rescue the phenotype by expressing rapamycin and reducing the overexpression or activation of TOR signaling. And indeed, you see there is a very nice rescue when you feed the flies rapamycin and, and probably decrease the pathogenicity. So what I've shown you is that through the tech cycle, these two proteins here, nardelysine chaperones this protein. This leads to an upregulation of alpha-ketoglutarate. This leads to an activation of mTOR. This leads to suppression of autophagy. And we think neurodegeneration, autophagy, suppression of autophagy has been tightly linked in many cases to neurodegeneration. And feeding the flies rapamycin blocks this and reduces and delays the phenotype. I'm going to end here and answer some questions. There's a lot of people that were involved in this project. Lipid droplets was done by a number of colleagues that have done the biochemical assays, and Lucy was the leader together with Ke. We have a superb metabolomics facility, which is really important because it allowed us to nail down the phenotype. The nardelysine project was mostly done by Wan He and Michael, a physician. The human geneticists have played a key role in this project. Michael Wangler, who's an MD, and Jim Lupsky, together with the people in Jim's lab. And for those of you, for the nardelysine, Valerie Arboleda and Stan Nelson here at UCLA. And I didn't talk about the Friedreich's ataxia story. Thank you.